So we will be starting this week's lecture. This is week 2 for microbiology. We will start off by briefly reviewing some of the concepts that we had gone over in the previous week's lecture. Again, this is a very brief review, and we may introduce some additional information over the previous week's topic. Now with that said, please be sure to fully review the first week's video. So we'll start off with the classification of organisms. And when we look over here, essentially what we see are that the microbial world consists of two areas. We have one area of living organisms, and we have another area of infectious non-living organisms. For the living organisms, we have the domains of the three domains being that of archaea, bacteria, and eukarya. And we said the oldest of these three would be that of archaea. We said that both archaea as well as bacteria, archaea and bacteria, they lead to prokaryotes, right? And prokaryotic organisms. And then we said that eukarya leads to eukaryote organisms, which include algae and protozoa, which contribute to protists. We also said eukaryotic organisms may be fungi. And we said that eukaryotic organisms could also be hominids, multicellular parasites. So remember, these are within the microbial world, microorganisms we're discussing. And if you notice, algae, protozoa, and fungi, these can be either unicellular or multicellular organisms. The exception being the hominids. These are multicellular parasites. So again, as he said, there are three categories called domains in biology. The first domain is archaea, which are like bacteria, but they have a different metabolism that allows them to live in extreme environments. The second domain is bacteria, which we can find everywhere, and they play an important role in many ecological processes. The third domain is eukarya, which have more complex and organized cells than bacteria and archaea. Eukarya can be either unicellular or multicellular, and they have a nucleus and organelles such as a mitochondria. So, when you have knowledge about these domains, it helps us to understand the diversity of life and and we're able to better address biological and environmental issues. So again, two types of cells, prokaryotic cells and eukaryotic cells. Prokaryotic cells, like those that are found in archaea and bacteria, they do not have a true nucleus. Instead, their DNA is located in a region of the cell that's called the nucleoid. Okay? So again, prokaryotes, their genetic information, their DNA is found in an area of the cell called the nucleoid. In addition to that, they also lack membrane-bound organelles that we find in eukaryotic cells. So again, a very big distinction between them is that they do not have membrane-bound organelles. Eukaryotic cells, on the other hand, they have a true nucleus that's enclosed by a membrane, and they also have membrane-bound organelles. They have many types of membrane-bound organelles. So again, as we looked at in the previous slide, some examples of these eukaryotic cells, they include protists, plants, fungus, and animals, right, such as the hominids. So this difference in cell structure has significant implications for the biology and the functioning of these organisms. When it comes to archaea, archaea are a type of prokaryotic cells that are similar in their size and appearance to bacteria. They're able to multiply by binary fission, and they may even move around by flagella, but the cell walls, they lack peptidoglycan, and their rRNA sequence is different. Many archaea are extremophiles, which means that they're able to survive in extreme environments such as very high salt concentrations or very high or low temperatures. And these differences make archaea a unique type of organism. And because of their uniqueness, it has very important implications for biology and ecology. What more we can say is that, again, these archaea, they're believed to be the oldest known organisms on planet Earth. Actually, their molecular structures are much closer to eukaryotes than bacteria. Some of these archaea, they've also even been found to colonize humans, but there isn't any evidence that they cause any disease. So when we look at bacteria, again, they are prokaryotic organisms. So prokaryotes are single-celled organisms that lack a membrane-bound nucleus or any other membrane-bound organelle. What does it mean when they lack? L-A-C-K, lack means they do not have. They have their DNA that is located in the nucleoid, as we said earlier. And most have a specific shape. They also have a rigid cell wall, which consists of peptidoglycan, and peptidoglycan is unique to bacteria. Prokaryotes, they multiply through binary fission, and many bacteria are able to move using flagella. So when you talk about eukaryotes, they have a true nucleus. They have membrane-bound organelles. They're much more complex than prokaryotes. As we said earlier, their family, they include fungi, algae, protists, even and animal organisms such as the helminths, or again, the worms. All right, moving on to algae. In summary, algae are a diverse group of organisms that can be single-celled or multicellular, and they're capable of photosynthesis. Algae primarily live in water, and they have rigid cell walls. And many of them, they have flagella, which is used for movement. The cell walls and flagella of algae are distinct from those of prokaryotic organisms. Moving on to fungi. Fungi are a diverse group of organisms that can be either single-celled or multicellular. They get their energy from breaking down organic materials, and also they play an important role in ecosystems as decomposers. Primarily, fungi live on land, and they're able to be both beneficial as well as harmful to other living organisms. They can be either single-celled, such as yeast, or multicellular, such as molds and mushrooms. Unlike plants and algae, fungi, they don't use sunlight to produce energy. Instead, they get their energy by breaking down organic materials such as dead plants and animals. This process is what we call decomposition. Fungi, they're They're very important decomposers in the ecosystem because they help recycle nutrients back into the soil for other organisms, for other living organisms to use. Primarily fungi, they live on land in a variety of habitats such as forests, fields, and even perhaps in the yards that some of you may have. They play an important role in the ecosystem, again, by their role in decomposing dead materials and breaking down nutrients. But as we had mentioned earlier, they can also be important pathogens that cause disease in plants, in animals, as well as us in humans. All right, protozoa. Protozoa are a diverse group of organisms that are single-celled and they are larger and more complex than prokaryotes. They get their energy by ingesting organic compounds, which means they eat other organisms. Unlike some of the other groups like algae or plants, protozoa do not have a rigid cell wall. They're mostly motile. Again, that means that they are able to move around. And again, they do this by using structures such as flagella or cilia. We can find them in a variety of environments, including freshwater, saltwater, soil, and even inside other organisms. Some of these protozoa, they're beneficial. Others, they can be harmful, and they can cause disease in humans and other animals also. So if you look over here, again, what are some of the key differences between procs and eucs? Okay, prokaryotic cells and eukaryotic cells. Generally speaking, procs are much smaller, okay, compared to eukaryotic organisms, whereas procs are always single-celled. Eucs, a lot of the times, they can be multicellular also. Again, they can be both unicellular or multicellular. We said that prokaryotes, they don't have a membrane-bound nucleus or other organelles, whereas their counterparts, they do have both conditions that are met. Usually they have a very distinct shape DNA. It tends to be circular without proteins. On the other hand, in eucs, the DNA is more linear. They have a linear DNA, okay? And it's associated with proteins to end up forming chromatin. There's also differences in the ribosomes. So in procs, there's smaller 70S ribosomes, whereas in eucs, there's larger 80S ribosomes. Procs do not have a cytoskeleton, whereas eucs always have a cytoskeleton. Prokaryotes, they may have a flagellum, okay, that's used for motility. Eukaryotes also have a flagellum, but again, the type is very different, right, between the prokaryotic flagellum and the eukaryotic. Very different makeup between the two. Procs will divide by binary fission, whereas eucs will divide by cell division, such as meiosis and mitosis. In prokaryotes, reproduction is always asexual. In eucs, reproduction could be either sexual or asexual. We also discussed in the previous lecture the Linnaeus system of scientific nomenclature as being the standardized way of naming and classifying living organisms. So it was created by a Swedish naturalist whose name is Carl Linnaeus. Okay, so Carl Linnaeus came up with this system back in the 18th century, and again, it's still used to this day. And in this system, each organism is given a unique two-part Latin name that consists of a genus and a species name. The genus name is capitalized, and both names are italicized or they could be underlined. So again, for example, in humans are known as Homo sapien. Okay, Homo being the genus, and sapiens being the species. Then we said we have the non-living members of the microbial world, right? So they could be viruses, viroids, or prions. Again, these are acellular infectious agents. Okay, they do not have a cell. We do not consider them to be living. So again, these are obligate intracellular agents that are tiny organisms that cannot survive outside of a host cell. Okay, so they need a host cell to survive and to reproduce themselves. All right, so here we go. Again, these are viruses and viroids. These are obligate intracellular agents. Right? This is one of the key things that you have to realize about them. Prions, on the other hand, again, these are just abnormal forms of cell protein. Okay? And they're just made up of protein. No DNA, no RNA, just, again, abnormal folds of protein. Okay. Let's talk about what an incubator is. Now, these are some of the equipment that you may find in a laboratory, a biology laboratory, a microbiology laboratory. You may find these equipment in the lab of a hospital where they're testing they may be testing blood or other types of body tissues. So first thing we look at is an incubator, and this is a tool that's used to grow and maintain microbiological cultures. And it's able to maintain, what an incubator will do is it maintains an optimal temperature, a consistent temperature at a specific humidity, and also it is able to maintain the proper gas range as well, of such as carbon dioxide and oxygen. Okay, that will be supportive to growing some microorganisms, some type of microorganism. So if you look over here, this is a picture of a incubator. Kind of looks like a refrigerator, but not the same. Or again, another way to look at it is you can think of it as a specific type of an oven. Of course, it doesn't get that hot, but again, it provides the conditions that are optimal for growth. Notice that there's not any light entering. Again, bacteria, they tend to like dark areas. Okay, what's an autoclave? And now an autoclave is a pressure chamber that's used to sterilize equipment and supplies by exposing to extreme pressure and temperature, as well as for a specific duration. Okay, so for a specific time, at a specific heat, in addition to specific pressure. The combination of these things ends up killing nearly all types of microorganisms. Right? So again, this is useful, for example, in the medical field before surgery. Right? If you're doing any type of a surgery or any type of a medical procedure, it's more economical to reuse medical instruments. So if you have, again, steel instruments, you can clean these, wash them with detergents, and after washing them with detergents, then you would wait for them to dry. After they are dried, then you would sterilize them in an autoclave. This is a picture of a couple of different type of autoclaves. Again, one kind of looks like a washing machine or a dryer, I guess, in shape. In that, there's a barrel inside, and inside this cylindrical chamber, you see a couple of shelves, and again, you can place instruments or whatever it is that you want to have sterilized inside. You close the door or the lid, and then steam is used to create pressure and high temperatures for a certain duration, for a certain period of time, so maybe 20 minutes, 25 minutes. And after that, then whatever the contents are inside, they would be void of any pathogenic organisms. Also in a microbiology lab, you'll find a centrifuge. And again, this is a machine that essentially turns very fast, maybe 5,000 times in a minute or 6,000 times in a minute. And essentially the role of this machine is to separate substances based on their different densities. So again, we use this very commonly for blood. Okay? After you draw blood from a patient, if you want to run a blood chemistry, for example, again, you will use a test tube, typically with a red colored top or even a yellow colored top, and then you'll place that test tube after you've drawn the patient's blood into this machine. Then this machine will spin maybe 5,000 times a minute or more for about 5 minutes, 8 minutes, okay, whatever the recommended time is that the manufacturer advises for the machine. After that time, then what ends up happening is when you look at the tube, the red blood cells, they fall all the way to the bottom, and the white blood cells, they sit on top of the red blood cells, and then on top of that we have the plasma, and then we end up testing the plasma for chemistry, okay, for electrolyte level, for glucose, for sodium, for certain hormones. So again, this is an interior view of this centrifuge, and you can see that you take these test tubes, you put them inside, and then they will turn, okay, very fast. And then again, whatever the contents of this are, they will separate based on their densities. This is a hot plate or a stir plate. So you take a flask or something else, whatever, maybe perhaps it's a test tube or another type of container, and you put it on top of over here, and what it does is two things. First of all, it heats up, okay, so it raises the temperature, so you can start to heat the solution. In addition to that, it will, you take one of these things, this is a magnet, and you drop the magnet inside, and what this machine will do is that it creates a magnetic field, okay, that oscillates, and that allows for this magnet to move around, and essentially what it's doing is that it's stirring the solution as well as heating it up. Right. So again, this is used in different types when you're trying to grow certain substances, or again, even in any type of chemistry experiments. In a organic chemistry lab, you will use these. All right, moving on to a Stowmaker. This is a kitchen tool that's used to mix or blend food samples for microbiological testing. It's made up of a plastic bag with a paddle-like structure that's inside that bag, and it moves back and forth to crush and homogenize the food sample. This instrument, we use it to prepare a representative sample of a food product for microbiological analysis. And what it does is that it ensures that any bacteria or other microorganisms in the food are evenly distributed through the sample. So this machine is commonly used in the food industry, and we also use it in research labs to study microbial contents of different foods. So what's a vortex? Right? It's a device that's used very commonly in labs to mix small vials or test tubes. That's it. Just kind of vibrates a lot and things start to get mixed. That's it. All right? That's all a vortex does. This is also very common that we find. A Bunsen burner. Right? What is this? Again, this is just kind of like a stove, okay, for a laboratory. What do we have here? Nothing fancy. This is a tube. It's connected to a gas source, and this is a regulator which regulates the amount of gas that's entering, and the gas ends up flowing through this tube through this regulator and then up this opening. It exits, the gas will exit from this point. And again, if you ignite this gas, you expose it to a spark or fire, it will light up. Now you can take some solution or whatever you want. If you want to heat it, you're able to heat it up over here, okay, on top of this flame. That's it. That's all it is. So we often use, for example, if we're going to be using, if we're streaking bacterial samples, after we streak a sample, we will take this inoculating loop and sterilize it on that flame of that Bunsen burner to sterilize, to make sure that all the bacteria are dead, okay, before we start to use it again. So again, these inoculating loops, we use this to transfer microorganisms to perhaps a growth medium, or a petri dish, or for staining slides onto a slide. And it's made up of a handle and a wire. Okay, it's a steel wire. And again, it can vary in diameter. Maybe it's 5 millimeters. So again, the metal, it's usually either platinum or nichrome. Right? These are the two common metals that are used, either platinum or nichrome. So again, you can see over here, these are various types of inoculating loops. And these are disposable ones, plastic ones. You can use it once and you throw them away. So you'd use this one, for example, and you take a sample and then you would streak it onto a petri dish. And after you streak it, then you would grow it. Perhaps you'd place it in the incubator that we looked at earlier. So in this example here, this scientist is streaking, or not streaking, she is introducing whatever that she took a sample from into this growth medium, which is a liquid broth. So again, in labs we use graduated cylinders, okay, again, which measure a certain amount of liquid, and if you notice, there's all different types of sizes. Also, pipettes are used to measure how much liquid you want to take out from somewhere. These are very precise instruments used to measure quantity of liquid. So again, when we looked at those other pictures, what we saw are various flasks, right? And you notice that they also come in very different sizes. And again, when these are used to heat or store substances. Also, many test tubes are used, and these test tubes, they also come in different sizes. Again, they are used to store, they're used to heat or mix substances, and we use this rack, a test tube rack, to hold these test tubes after they're filled. Otherwise, they'll fall down, they'll roll over. All right, so that's the limit that we're going to be looking at these lab equipment. Moving on with cells. Cells, the word cell comes from the Latin word cella, which means a small room. And as we had mentioned in previous lectures, and in other courses also, as well as your anatomy class and your physiology, you should have learned and you should know by now that cells are the smallest unit of life. They are the building block of life. Right? They have the capacity to replicate independently also. And cells are of two types, eukaryotic cells and prokaryotic cells. Eukaryotic cells, they have a nucleus. Prokaryotes do not have a nucleus. Prokes, or the prokaryotic cells, comes from the Greek word for pre-nucleus, right? Before the nucleus. Eukaryotes comes from the Greek word for true nucleus, okay? Prokaryote, eukaryote. So again, true nucleus before the nucleus. Remember, prokes do not have a nucleus. Again, when you look over here, this illustration is showing you a prokaryotic cell as well as a eukaryotic cell. Notice the diversity between the two. Okay? Be sure you study this picture and you look at the difference. Look at the size difference. Prokes are very small compared to eukes, which are much significantly larger. Also, if you look at the makeup of the organelles that we have inside eukaryotes, extremely diverse, and again, they're complex compared to that of the eukaryote. Right, looking at this picture of a prokaryotic cell, notice first of all, if you look over here, notice that the nucleoid, okay, we have circular DNA. Right? There's no nucleus that's present. The ribosomes also are different than that of the eukaryote. Remember, they have a 70S ribosome compared to the 80S in the eukaryotic cells. Also notice that, again, this is relatively simple inside. There's not much going on over here aside from DNA and the ribosome that we have over here. The cell wall, okay, notice that this will be different. There's also a capsule, all right? So, all right, so bacteria that are monomorphic maintain a constant shape, size, and form throughout their life cycle. On the other hand, pleomorphic bacteria are variable in their shape, size, and form, and they're able to change their appearance under specific conditions. So if you look over here in terms of some of the basic shapes, we have a bacillus. So this is kind of like a capsule-like, okay? You have a diplococci, and this is two spherical, okay? Di means two, okay? You have these two spheres, diplococci. Spirillum, if you look over here, it spirals, whereas a spirochete is more of a helical structure, okay? So this is more of a spiral, and this is more of a helical structure. Then if you look at a vibrio, this is more kind of like a comma-shaped. We can also have some irregular shapes, such as a star. If you look over here, so sometimes you'll see these star-shaped bacteria. Other times you may also see rectangular-shaped bacteria. Now, in terms of their arrangement, so in the previous slide, we looked at these circular ones, right? We said we're cocci. Di meaning two. And then if you look at these rod shape, we have diplo, right? Two bacilli, which are the rod shape. So diplobacilli, diplococci. Or we can have clusters. And these clusters, we say they are staph, right? Staphylococci, clusters of spherical cells. Or we can see them in chains, right? We call these strep, right? So streptococci, because these are spherical. So this means the shape, and this is the arrangement. Or they can be in a chain or a series of rod-shaped, so streptobacilli. And again, this is another representation of the different shapes and arrangements of cells. Be sure you explore these and look at these, as we've already discussed the majority of these in the previous slides. Okay, capsules and slime layers are structures that surround some bacteria. Their general function is to protect the bacteria from the host's defenses, as well as to enable the bacteria to adhere to specific surfaces. Capsules, they tend to be thick, rigid, and smooth, while the slime layers, they're thinner, they're less rigid, and they're more globular. The chemical composition of capsules and the slime layers will vary depending on the bacterial species, but most are made up of polysaccharides. Together, capsules and slime layers, they're referred to as glycocalyx. Okay, so capsules and slime layers together are referred to as glycocalyx. And this word, literally, if you break it down, it means sugar shell. Okay, glyc means sugar, calyx means cell. Sugar cell. Sugar shell. Okay, not cell, sugar shell. All right, so after that tongue twister, sugar coat, okay? We can say that the glycocalyx is a layer of sugars or polysaccharides that surrounds the outer surface of the cell membrane. And when you look at it over here in an image that's taken from an electron microscope, we see that it consists of polar oligosaccharide side chains that are covalently linked to proteins and some lipids of the plasma membrane. In terms of the function, the glycocalyx has various functions, such as protecting the cell from its environment, helping the cell adhere to surfaces, and it also helps identify the cell to the immune system. The thickness and the composition of the glycocalyx will vary depending on the type of cell and the environment that you find it. Alright, now we're going to move on to flagella and pili. So some bacteria have these appendages that are made up of proteins. They're not essential for living, for life. They do help, they may help it survive in certain environments. Okay, so let's take a closer look at these. Alright, so we'll start off with flagella. And flagella are long protein structures that are responsible for the motility of certain types of bacteria. They're made up of chains of flagellin and use a propeller-like movement to push bacteria forward. Flagella are able to rotate more than 100,000 revolutions per minute. So that literally translates into speeds of about 130 kilometers an hour. Some bacteria, they will use their flagella for pathogenesis, such as H. pylori, Helicobacter pylori, which uses it to penetrate through the mucus coat. Flagella we can see with the aid of a microscope, and again, they're important for the identification and classification of microorganisms, of bacteria. So looking over here, we see that flagella structure has three basic parts. There's a filament, a hook, and a basal body. Okay, filament, hook, and basal body. The filament, it extends to the exterior, is made up of a protein called flagellin. The hooks, they connect the filaments to the cell, and the body, it anchors the flagellum to the cell wall. Bacteria will use flagella for moving, for motility, and again, this allows them to move towards nutrients, a food source, or it can help them move away from some harmful compound. Now, this movement is known as chemotaxis. Okay? Bacteria can sense chemicals in their environment, and if the chemical is a nutrient, it acts as an attractant. However, if the chemical is toxic, it acts as a repellent. The rotation of the flagella is responsible for the run and tumble. Okay? The run and tumble movement for bacteria, and this is what's allowing them to move towards or away from the chemical compounds in their environment. Alright, so you can see the various arrangements of these flagella. Sometimes you find them only on one side and there's only a single flagellum that you see over here. So we call that monotrichous and polar. Other times it's peri. Peri means around. Peritrichous. So we find multiple flagellum surrounding the bacteria. Lophotrichous and polar. Again, you have multiple flagella on one pole. And then amphitrichous and polar, we find them on both poles. We have a single flagellum on both poles, both sides. So again, this slide is just going over what we looked at in the pictures in the previous. So be sure you review this and you have a good understanding of them. On this slide, this is just an illustration that's showing you the motility of cells, okay, with the help of the flagellum. So again, the flagella, they rotate to either run or tumble Okay, so move toward or away from their stimuli. This is what we're looking at, the run and tumble, based on the direction when you look at the rotation of the flagella. So the axial filaments, which are also called the endoflagella, and you can see over here in the spirochete, they're anchored at one end of the cell, and the rotation is what's going to cause the cell to move. So again, in this picture, again, you're just looking at the diagram of these axial filaments. All right, pili and fimbria. So pili and fimbria are both hair-like structures that are found on the surface of bacteria, but they differ in their function and location. Fimbriae are short, thin, and numerous, and they are used for attachment to the surface, such as the host tissue or other bacteria. Okay, so fimbriae, they allow for attachment. Okay. Now, on the other hand, pili are longer, they're thicker, and they're far fewer in number, and they're involved in bacterial conjugation. Okay, so pili are involved in bacterial conjugation, and this is a mechanism for genetic exchange between bacteria, right? Pili also play a role in attachment, but their main function is to transfer genetic material from one bacterium to another. So, in summary, fimbriae, they're used for attachment, while pili can also be used for attachment, but their primary purpose is for conjugation, right? For transferring genetic information. Again, this is showing in this slide, pili are longer, they're far fewer than the fimbriae. Okay, fimbriae, again, what are they for? We find them much greater quantity, and also they are used for attachment. All right, so looking over here in this slide, what is it saying? It's saying that the cell wall prevents osmotic lysis. Why? Because in bacteria, the cell wall is made up of peptidoglycan. All right, so what is peptidoglycan made up of? What's its makeup? And it consists of two monomers. Okay, so this polymer, okay, it's a disaccharide, consists of two monomers. One is N-acetylglucosamine, and the other is N-acetylmuramic acid. All right, now we're going to be looking at gram-positive and gram-negative cell walls. So gram-positive and gram-negative cell walls are two different types of bacterial cell walls that differ in their structure and composition. The gram-positive cell wall is made up of a thick layer of peptidoglycan. They will appear purple when we stain it with crystal violet and iodine in the gram staining process. Gram-positive bacteria, they have a simpler cell wall structure with fewer layers. They lack an outer membrane, and they have teichoic acid and lipoteichoic acids embedded in the peptidoglycan layers. Now, when you compare that with a gram-negative cell wall, they have a thin layer of peptidoglycan that's surrounded by an outer membrane. The outer membrane provides additional protection and acts as a barrier against antibiotics and the host's immune defenses. Gram-negative bacteria will appear red or pink when we stain it in the gram staining process. So the differences in cell wall structure and composition plays a very significant role in the susceptibility of the bacteria to different antibiotics, as well as their ability to cause disease. So over here, this is just showing you the differences between the gram-positive and the gram-negative cell walls. Again, we had discussed some of these differences. One thing that they mentioned that I didn't mention in the previous slide is that in the gram-positive, there's no outer membrane. And in the gram-negative, again, there is a periplasmic space. Okay, so be sure you're familiar with these terms, periplasmic space, which is found in a gram-negative cell wall. So, teichoic acids are a class of molecules that are found in the cell walls of gram-positive bacteria. They're made up of a long chain of sugars and are divided into two types: lipoteichoic acid and wall teichoic acid. Lipoteichoic acid is anchored to the plasma membrane of the bacteria and extends outwards, while the wall teichoic acid is linked to the peptidoglycan layers of the cell wall. These molecules have various functions that include regulating the movement of cations, providing structural support to the cell wall, and providing antigenic variations to help the bacteria evade the host's immune system. Looking at a gram-negative outer membrane, we have lipopolysaccharides, or LPS, lipoproteins, and phospholipids that forms the periplasm between the outer membrane and the plasma membrane. So the LPS, the lipoprotein, and the phospholipids are components of the bacterial outer membrane. These molecules collectively, again, they form the periplasm. And remember, this is the space between the outer membrane and the plasma membrane. The other thing that we can say is that the lipoproteins are the proteins that anchor the outer membrane to the peptidoglycan layer. And functionally, the phospholipids are the main structural components of the plasma membrane. And they also contribute to the fluidity and the stability of the membrane. The LPS, we can say, plays an important role in protecting the bacterial cell from certain antibiotics as well as the host's defenses. So the gram-negative outer membrane provides protection to the bacteria from phagocytes, from complements, as well as antibiotics. It contains important components such as O-polysaccharide antigen, which is used for the identification of bacterial strains, and lipid A, which is an endotoxin that can cause harmful effects in humans. Porins, which are proteins, form channels through the membrane, allowing for the passages of molecules. Okay, mycoplasma is a type of bacterium that causes mild pneumonia, and it's unique in the sense that it lacks a cell wall. This feature makes antimicrobial treatments that are directed towards the cell wall ineffective against mycoplasma. So instead, the strength of mycoplasma's membrane comes from the presence of sterols. Okay? Bacteria in the domain Archaea also have a variety of cell wall types, but they do not contain peptidoglycan. Right? Instead, they contain pseudomerins, which lack, as we'd mentioned earlier, the NAM and the D amino acids. All right, let's talk about the Gram stain mechanism. So the Gram stain mechanism is a laboratory technique that's used to differentiate bacteria into two groups, gram-positive and gram-negative, based on their cell wall structure. So the process involves adding crystal violet iodine to the cells, which forms crystal complexes in the cell wall. In the next step, an alcohol solution is used to dehydrate the peptidoglycan layer. What we see resulting in gram-positive bacteria is that the alcohol doesn't affect the crystal complexes, and this results in the cell staining purple when we look at it. In contrast, the alcohol dissolves the outer membrane and leaves holes in the thin peptidoglycan layer in gram-negative bacteria. As a result of that, the crystal complexes wash out, and the cell appears pink after we counterstain it with safranin. So if you look over here, we see purple stain cells, okay, which would be gram-positive, and over here we see these pink stain cells, which are gram-negative. All right, moving on to the plasma membrane. So you can see over here is that the plasma membrane is made up of a phospholipid bilayer. Okay, one layer facing the outside of the cell, another layer facing the inside of the cell. Embedded within this phospholipid bilayer, we have some proteins that are transmembrane. They span across both layers. Okay, some of these proteins are called integral proteins. They're stuck within part of this membrane, right? Other proteins are peripheral proteins. They may be attached to just one side of the protein, perhaps the outside or perhaps on the inside membrane. We also say that this plasma membrane has this fluid mosaic model. This means, again, the membrane it's always moving, okay? It's literally going from side to side, side to side. These phospholipids, they're constantly moving at all the time, and this provided fluidity. We say that the plasma membrane is selectively permeable, meaning that it allows certain things to enter while it prevents other things to not enter. Or we can say that it allows certain things to exit while it prevents other things from exiting. So again, this is the property of it being selectively permeable. Photosynthetic pigments are pigments that absorb light energy and convert it into chemical energy during photosynthesis. In some photosynthetic bacteria, these pigments are found on foldings of the plasma membrane called kinetophores or thylakoids. Chromatophores are membranaceous structures that are found in some bacteria, algae, as well as plants. They contain photosynthetic pigments such as chlorophyll as well as carotenoids, which are used for photosynthesis. The pigments, they're found arranged on foldings of the chromatophore or thylakoids, and this increases the surface area that's available for light absorption. The pigments absorb light energy, which is then used to drive the process of photosynthesis, converting light energy into chemical energy in the form of ATP and NADPH. In some bacteria, such as purple and green photosynthetic bacteria, chromatophores also play a role in generating proton motive force that can be used to drive ATP synthesis. Right, bacterial cells have a variety of internal structures. Some of these structures are essential for life, such as chromosomes and ribosomes. Others are optional, and they may allow certain bacteria to have advantages if they have them. So things like endospores, storage granules, or plasmids. So when we're looking at the protoplasm or the cytoplasm, we're talking about the area inside the bacteria where its genetic information is going to be found, okay, where its ribosomes are going to be found. So again, this is providing an area. It's the medium where the chemical reactions, the synthetic processes are taking place. Okay, so when we're talking about cytoplasm, again, we're talking about the area inside the plasma membrane. So this area is the cytoplasm. The nucleoid is the area where we have the genetic information. Okay, so this area over here, this would be called the nucleoid because this is where the genetic information is found. Ribosomes, these are small structures that are made up of RNA and proteins that are involved in the process of protein synthesis. Okay, this is a job for ribosomes. As I had mentioned earlier, the ribosomes found in prokaryotes are different than that of eukaryotes. In prokaryotes, we have a 70S ribosome that's formed, which is made up of two subunits. It is made up of a small 30S subunit and a larger 50S subunit. So the combination of the 30S and 50S produce a 70S ribosome. Okay, inclusions or granules are areas where nutrients are concentrated. So if you go over here, we have an inclusion over here. Okay, now you can see granules over here. Okay, so again, these are areas where nutrients are concentrated. So if you look at over here, we can see that, look, there's metachromatic granules, some of these inclusions over here, lipid inclusions, sulfur granules, carboxysomes, gas vacuoles, energy reserves. So these are all examples of inclusions. Endospores are a type of dormant structure that are produced by certain species of bacteria, such as Bacillus and Clostridium. These resting cells are highly resistant to various environmental stressors, including desiccation, heat, as well as chemicals, and this allows them to survive very harsh conditions. Endospores are formed through a process called sporulation, and they can remain in this state for extended periods of times until the conditions become favorable for germination and return to a vegetative state. So this unique ability to form and withstand endospores makes these bacteria of significant interest in both basic research as well as some of the applied fields such as food safety, as well as even in areas such as bioterrorism defense. All right, moving on with plasmids. Plasmids are small, circular, double-stranded DNA molecules that exist independently of the chromosomal DNA in bacterial cells. They can be transferred from one bacterium to another through horizontal gene transfer, and often they carry genes that provide the bacterium with additional traits such as antibiotic resistant, virulence factors, as well as metabolic capabilities. Plasmids can replicate independently and quickly within the bacterial cell, and this allows for rapid adaptation to changing environmental conditions. Okay, this is a eukaryotic cell. Okay, so again, one of the defining characteristics is that this has membrane-bound organelles. It has a membrane-bound area where the genetic information is found. Okay, so over here you can see this is the nucleus. Here's the nucleolus. You can see the DNA over here also. Okay, and then we see over here we have endoplasmic reticulum that's found. We see a Golgi apparatus that we have. We find mitochondria. We have, of course, we have the plasma membrane over here. This area is the cytoplasm. All this area over here, within the cytoplasm, we have again, all these are the organelles, including centrioles, centrosomes, lysosomes, peroxisomes, proteasomes. All these are organelles. These are small tools that we find within the eukaryotic cells. And remember, these are complex cells compared to prokaryotes, and they're much larger cells compared to prokaryotes. So we can say that the cell membrane in the eukaryotic cell, it's made up of phospholipid bilayer that has proteins embedded in it that perform various functions such as transport, cell integrity maintenance, attachment of proteins to its internal structures, and also it acts as receptors for cell signaling. The membrane also contains sterols for strength. With animal cells, it contains cholesterol, and fungal cells contain ergoesterols. Receptors in the outer layers are typically glycoproteins. So overall, the cell membrane acts as a barrier that separates the cell from its environment while allowing necessary molecules to pass through. Again, this we call it what? This is the property of it being selectively permeable. Endocytosis is a process that occurs in eukaryotic cells to bring in materials from the surrounding environment. The most common type of endocytosis in animal cells is pinocytosis, where the cell pinches off a small portion of its own membrane along with attached materials such as fluids and solutes. The resulting vesicle is internalized and is called a endosome. So the process of endocytosis allows the cell to take in the necessary nutrients and other molecules that may not be readily available in the extracellular environment. Phagocytosis is a specific type of endocytosis that is important in the body's defense mechanism. In this process, a phagocyte cell, such as a macrophage, sends out pseudopods that surrounds the microbes. The phagocyte then brings the microbe into a vacuole called a phagosome. Okay? The phagosome then fuses with a lysosome, which is a sac of enzyme and toxins, to form a phagolysosome. Okay? The microbe dies in the phagolysosome and its material is broken down by the enzymes in the lysosome. This process is crucial for the immune system to eliminate harmful microorganisms. Exocytosis is the opposite of endocytosis. Okay, so the vesicles inside the cell fuse with the plasma membrane and it releases the content into the outside, okay, into the external environment. All right. Eukaryotic cells are characterized by several unique structures that differentiate them from their prokaryotic counterparts, right? From prokaryotic cells. One of these structures is a cytoskeleton, which provides shape, support, as well as movement to eukaryotic cells. The cytoskeleton is made up of three types of protein fibers. There's microfilament, intermediate filament, and microtubules. Right? Eukaryotic cells also have flagella and cilia, which are used for motility and movement. Flagella, remember, we looked at them before in prokaryotic cells also, but again, these are very different in eukaryotic cells. So, remember, flagella allow for movement, right? They are whip-like structures that move in wave-like motion. Cilia, on the other hand, are shorter. They're hair-like structures. Okay, they move in a very coordinated wave-like pattern. Okay, so both structures are made up of microtubules and are anchored in the cell's basal bodies. Additionally, as you had mentioned earlier, the ribosomes are different in eukaryotic cells also. Eukaryotic cells have a larger, more complex ribosome known as the ADS ribosome. These ribosomes are made up of, this ADS ribosome are made up of 4 types of RNA and around about 80 different proteins compared to the 70S ribosome that we found in the prokaryotic cell. So the larger size of the ADS ribosome allows for greater protein synthesis capability, and also it allows for the eukaryotic cell to produce a much diverse profile of proteins. Right, so in this slide over here, again, this is just showing the cytoskeleton. And remember, as we said, in eukaryotic cells, we can find three elements: microtubules, actin filaments, intermediate filaments. In this example that they've given over here for the cytoskeleton. Microtubules, again, these are the thickest of the cytoskeleton structures. These are long, hollow, cylindrical in shape, and they are made up of subunits called tubulin. Okay? They form mitotic spindles, and these are the main structures in cilia as well as flagella. Actin filaments are made up of actin polymers, okay? And it enables the cytoplasm to move. If you remember, we also find actin in muscle, right? These are strong molecules. Intermediate fibers, again, they function to strengthen the cell, and they also enable the cell to resist physical stress. Okay, flagella and cilia are complex structures that are found in eukaryotic cells. Flagella are long, they're whip-like structures that function in cell motility. Right? They have a flexible structure with 9+2 arrangement. This is 9 pairs of microtubules surrounded by 2 individual microtubules. Okay, so 9 pairs plus 2 individual microtubules. Now, on the other hand, cilia are shorter and more numerous than flagella and are often found covering the surface of the cell. Cilia can move the cell or propel the surroundings along a stationary cell. Now, both flagella and cilia, they play an important role in the movement of cells and the environment around them. So here you can see that, again, this flagellum, right, can move this entire cell. And over here, the cilia, it may move the cell, or it can move, again, the contents around the cell. It can move it, it can bring it close to it, or it can push it away as well. Okay, so it will have an effect on its surroundings, on the environment surrounding the cell. All right, so eukaryotes, they have many organelles. Okay, and remember, organelles, this term literally breaks down into L meaning small, organ means tool, small tools, right? And again, these membrane-bound organelles is another distinguishing factor between eukaryotes and prokaryotes. So what do we see? We see lysosomes, peroxisome, Golgi apparatus, endoplasmic reticulus, mitochondria, chloroplast in plant cells, and nucleus. Okay, so when we look at this picture over here, again, what do we see? The most prominent part of the cell would be this area here, right? This is the nucleus. This is where the genetic information is stored, all right? It contains DNA, right? This is where DNA can replicate in eukaryotic cells, either through a sexual process, meiosis, and also through asexual process, referred to as mitosis. Again, over here, they're showing you a picture. This entire structure is that of a nucleus, and again, it is bound by, it's a double 2-layer membrane, okay, that surrounds this, that it is made up of, that is protecting the area inside. And this prominent area within the nucleus is called the nucleolus. Okay, this prominent area is the nucleolus. And then around it, we have chromatin. Okay, this is this is the DNA over here. Okay, the DNA, the genetic information is found over here. That's the chromatin. The nucleolus, this is primarily we're looking at the building blocks. This is where RNA ends up being synthesized. Okay, this is the area, all the ingredients, the building blocks that are needed for synthesis of RNA to take place. This is this part. This is how important RNA is. Moving on to ribosomes. So again, ribosomes, we can say we have, there are many types of ribosomes. Among some, what's written over here would be rRNA, or ribosomal RNA, right? and they are important in the synthesis of proteins. So again, they play a role in translation. We take the mRNA transcript from the DNA and produce proteins from that. And ribosomes we find both in prokaryotic cells as well as eukaryotic cells, even though they are different. As we said, in prokaryotes we have a 70S ribosome, and in the eukaryotes we have an 80S ribosome. Again, as I mentioned in the previous slide, in prokaryotes we have 70S ribosome, in eukaryotes 80S ribosome. We find the 80S ribosome in eukaryotic cells attached to the endoplasmic reticulum. In the cytoplasm, they are free floating. The 70S, again, we will find them in the chloroplast as well as mitochondria in the prokaryotic cells. Looking at the endoplasmic reticulum, these are structures, these are membranous, that form a membranous system of tunnels and sacs. These endoplasmic reticulum, again, this essentially is an extension of the nuclear envelope. Okay, this double-layered membrane that is part of the nucleus. As that membrane leaves the nucleus, it extends into, again, this system of tunnels and sacs. When we find ribosomes attached to them, we call it the rough endoplasmic reticulum. When we do not find any ribosomes attached to them, we call it the smooth endoplasmic reticulum. Okay? So the rough is where we have protein synthesis that takes place. The smooth is where we have lipid synthesis. And also we have storage for calcium. Golgi apparatus, this is the structure that is kind of like a post office. Okay, it's sorting and packaging proteins, and then it'll deliver. It'll send out to deliver. Okay, that's the job for Golgi apparatus. The mitochondria, on the other hand, is also it's a double membrane organelle, and this is the site for energy production, okay, or ATP is produced. Also, the mitochondria contains DNA. Okay. Chloroplast we find only in plant cells. We do not find these in animal cells. Okay. Plant cells as well as algae. And this is a site of photosynthesis, and also this is a double membrane organelle. All right, so going back, reviewing what we have looked at a little bit earlier. Cell wall, again, this is an outermost layer surrounding the plasma membrane of many organisms such as plants, algae, and fungi. We do not have it in animal cells. All right, so it's made up of various carbohydrates, including cellulose, chitin, glucan, and mannan. The carbs provide support as well as protection for the cell. Now, in animal cells, okay, the outermost layer is the glycocalyx. It's made up of carbohydrates that are extending from the plasma membrane, and it ends up being bound to proteins and lipids in the membrane. And as we said earlier, the glycocalyx plays a role in cell recognition, signaling, as well as protection. Okay, in the cytoplasm, right, this is the area inside the plasma membrane, right, cytoplasm. All right, so what do we have? The cytoplasm membrane, okay, it's the substance inside the plasma as well as the nucleus. Cytosol, this is the fluid part of the cytoplasm. Cytoskeleton, this is the framework that's holding up the plasma membrane. Okay, so it's made up of microfilaments, intermediate filaments, and microtubules. And there's also cytoplasmic streaming. This means that there's movement of substances throughout the cell. Okay, streaming. Okay, lysosomes and peroxisomes. Lysosomes and peroxisomes, they're both organelles that are found in eukaryotic cells, but they have different functions. Lysosomes contain enzymes that break down cellular waste and debris, as well as foreign materials that may have been taken in by endocytosis. They're also involved in breaking down cellular components that are no longer needed, such as organelles that have reached the end of their life cycle. Peroxisomes, on the other hand, are involved in breaking down fatty acids and amino acids, as well as detoxifying harmful substances such as alcohol and formaldehyde. They also play a role in the synthesis of certain lipids. So, while both lysosomes and peroxisomes contain enzymes, the type of enzymes and their functions are what's different. Alright, so we said the nucleus contains the genetic information. The ER, we said it's an extension of the nucleus. It's either going to be rough or smooth. We also discussed the Golgi complex, its role, its purposes. It's that it is a membrane formation, and it's going to be secreting proteins. In other words, it's going to be exporting it after it sorts and stores it. We also discussed the differences between lysosomes as well as peroxisomes. We spoke about vacuoles, okay, that we have that we find in these cells, and again, we talked about its role in support as well as bringing foods into the cell. We talk about mitochondria and their role in producing energy. In plant cells, we had said that chloroplasts are where photosynthesis takes place. And then centrosomes, we also spoke about function of these centrosomes. Again, is to organize microtubules in the animal cells during cell division. Alright, so that's it for this lecture. Thank you.